Good day, class, and thank you for attending our first lecture. So, for our first meeting, I'm going to discuss to all of you our first lesson, the introduction to mycology. So, this is a very important aspect for you to be able to understand the study of fungi known as mycology. So, with that, let's begin. Now, here are the learning objectives for this lesson. You're going to know the general features and characteristics of fungi, identify the different ways on how fungi class is being transmitted. You're going to learn about the methods of collection, transportation, and handling of different specimens for fungal tests. Then you're going to enumerate the laboratory tests for identification of the different fungal infections known as your mycosis. And you're going to state ways on how fungal infections could be prevented and could be controlled. So from here on, class, please use your handouts, fill up the blanks, and take note of the different additional information that I would be stating. Now, mycology class, please take note, refers to the study of fungi, which are eukaryotic organisms that are involved in tandem with the animal kingdom. Now, before I continue with this slide, let's have a recall. Your bacteria class in your bacteriology, if you still remember, your bacteria is known as what type of organism? I'll give you a few seconds to answer. I'll call someone or anyone could volunteer. So bacteria class are basically prokaryotic. If naaalala nyo pa, they are prokaryotic microorganisms. They are prokaryotic microorganisms, and one of the key features key features of prokaryotic organisms is that they would divide by fission, specifically what we call binary fission. Di ba tinanong sa bacteriology niyo or na-discuss sa bacteriology niyo, how do bacteria replicate or reproduce? They would reproduce by binary fission. While your eukaryotic organisms class, they would usually reproduce via what we call mitosis. If you still remember, di ba sa mitosis, there would be a sperm, there would be an egg cell, and they would fuse or they would combine. So, ang sperm nyo is considered a haploid. Half meaning one half would contain the genetic material, so they would fuse together, forming a combination. So that's your mitosis, sperm and egg for most eukaryotic organisms. Now your fungi, they would divide via mitosis or something like that. So do take note of that. Let's continue. So this is the appearance class of your fungal cell or your fungal cell. So it has a cell wall. It also has a nucleus, a cytoplasm, a chromosome, a plasma membrane. And within the cytoplasm, you would be able to find the mitochondria, the one that envelopes the nucleus, the nuclear envelope. You could also find the Golgi apparatus and the plasma membrane. So I won't go much into details regarding the different organelles. I'm sure familiar naman kayo. But there's one thing that you need to take note of is that is this polar bud scar. This polar bud scar class is this is one of the key characteristics of a fungal cell. Later, I will explain ano yung polar bud scar na yan. Now, These are the different characteristics of your fungi class. Now, most fungi class, take note, are considered non-motile, meaning they cannot move. And they would possess a rigid cell wall with chitin. Now, another recall question in your bacteriology. Your bacteria class, in their cell wall, in their cell wall, what can be found? So if in fungal cells you can find chitin, what about in your bacteria? What can be found in your bacterial cell wall? So the answer to this class, to this recall question, is your peptidoglycan. If you remember, your peptidoglycan is responsible for the gram positive and gram negative of a bacteria. If it has a thick peptidoglycan, it's gonna be gram positive. But if it's thin or no peptidoglycan, it would be gram negative. So do take note of that. Your bacteria would contain peptidoglycan, but for fungi or fungal cells, they would contain chitin. Another characteristic of your fungi class is that they are non photosynthetic. Now, let's have another recall question regarding your biology or whatever life science subject you took before. Let's talk about your photosynthesis. Now, can anyone tell me what uh, what component or substance would allow photosynthesis photosynthesis to occur? So let me repeat the question. What component or substance would allow photosynthesis to occur? So to answer this class, ang photosynthesis nyo, if you're familiar, di ba, they would usually occur in plant cells. In plant cells, they have an organelle known as your chloroplast. And inside that chloroplast, you would find the substance chlorophyll. Chlorophyll would allow for photosynthesis to occur, generating energy. Diba? Ang photosynthesis nyo would be used in creating energy for the plant cells. So take note, class, that if we are going to compare fungal cells with plant cells, fungal cells are non-photosynthetic. The reason is because they have no chlorophyll. So that's one way to differentiate fungal cells from or fungi from plants. They are non-photosynthetic, they are non-motile. Now they cannot perform photosynthesis, which differentiate them from plants. The reason for this is because they do not possess chlorophyll, which I mentioned earlier. Now another characteristic class is that each fungal cell has at least one nucleus. So this is a uninuclear microorganism. And the nucleus is covered or surrounded by a nuclear membrane. It also has an endoplasmic reticulum, a mitochondria, and secretory apparatus or the Golgi apparatus. Now, most fungi are obligate or facultative aerobes. Now, another question, if you remember in your bacteriology, can anyone tell me what does facultative aerobes mean? Ano ang ibig sabihin yan? What does it mean, facultative aerobes? So when you say facultative aerobes class, they may or may not require oxygen to survive. So even without oxygen, even without oxygen, they could survive. While the word obligate, if we try to use a synonym for obligate, it would be required. So most fungi class they would require oxygen. Some may or may not require oxygen. So take note of that. So fungi are obligate or facultative aerobes. Now they are also considered chemotropic. Now from the word chemo would refer to somewhat requiring or producing chemicals. An example of a chemical they would produce class are enzymes. Enzymes that would degrade a wide variety of organic substances into soluble nutrients that are then passively absorbed or taken into the cell by active transport. So they use this substances class or chemicals. So the fungi would secrete these enzymes. They would degrade it or they would destroy the organic substances. And these organic substances are turned into nutrients, which are then absorbed and then taken via the so-called active transport. So that's one way on how your fungi would be able to absorb nutrients. And similar with your bacteria class, your bacteria can also secrete enzymes. If you still remember the enzyme streptokinase, streptolysin, these are the enzymes produced by your streptococcus that would allow for their pathogenicity. So, yan yung way ng mga bacteria nyo on how they can infect the cell. But for fungi, most of the time they use their chemicals or their enzymes to use it for as a source of nutrition. Now let's talk about the polar bud scar. So as I mentioned, one key characteristic of fungi is that they would have a polar bud scar. Now polar bud scar class is produced from a mother yeast cell. When it undergoes a type of asexual reproduction known as budding. Once the daughter yeast cell detaches, it would leave a scar. Now, from the word budding class, budding, ang yeast cell nyo, your yeast cell would be spherical in appearance. Now, they would usually reproduce. One way of reproduction is via asexual reproduction, known as your budding. What happens, class, is that this fungal cell, let's call this the mother cell. The mother cell class would reproduce via budding wherein it would produce another fungal cell. Now, once this fungal cell, itong fungal cell na to, this fungal cell would leave or detach from the mother cell. So, drawing ko ulit ha. I apologize for the terrible drawing. So, yan yung mother cell. Nag-detach na. Nag-detach yung na-produce na yeast cell, another yeast cell. It's going to leave a scar. Magli-leave siya ng scar. And that's a sign that the yeast cell was already able to reproduce. And this is an asexual reproduction known as your budding. This is different class from binary fission kasi 'di ba sa binary fission nahati siya into two. This one it produces another type of cell. Kaya nga siya, 'di ba sabi ko sa inyo, they would reproduce via mitosis. But this is an asexual reproduction. Basically from a single haploid. Wala kang there is no egg cell, there is no sperm cell. You just reproduce on your own, but not by fission or dividing, but instead you reproduce another daughter cell. So that's your polar bud scar. Let's continue. Now, what are the difference of your fungal cell with bacteria and plant cell? So, fungal cells have rigid cell wall external to the cytoplasmic membrane. Cytoplasmic membrane is made up of sterol. Sterol class is a type of lipid. If you still remember in your clinical chem, and to be specific, the sterol that makes up the cytoplasmic membrane of your fungi is known as your ergosterol. While in mammalian cells, the predominant sterol is cholesterol. So take note of that. So kapag cytoplasmic membrane, if asked what is the main component of the cytoplasmic membrane of fungi, the answer would be ergosterol. If asked what would be the main component of the cytoplasmic membrane of mammalian cells, it would be cholesterol. Now, fungi are usually haploid in their DNA content. Diploid nuclei are formed through nuclear fusion in the process of sexual reproduction. Fungi cell wall does not contain peptidoglycan. So as I mentioned, there is no peptidoglycan. Instead, they would contain class chitin. So there's no peptidoglycan, there is no glycerol, there is no teichoic acid, there is no lipopolysaccharide. Naalala nyo if you remember this lipopolysaccharide, this is a very important component of your bacilli. Instead, ang fungal cells nyo, instead of containing peptidoglycan, lipopolysaccharide, teichoic acid, glycerol, they would now contain carbohydrates, specifically complex polysaccharides. So instead of lipids, para mas matandaan nyo, for easier remembrance, just remember that if it's bacteria, the cell wall would contain lipids such as your peptidoglycan and lipopolysaccharide. But for your fungi, they would contain carbohydrates such as your mannans, glucans, and chitins, as well as structural proteins. So these are the differences between your fungal cell with your bacteria and your plant cell. Now, what are the general features and characteristics of your fungi class? Now, fungi that would cause human infections class are divided based on their morphological form. Now, in our subject in mycology, we are mostly tackling human infections class. Fungi that causes human infections. And there are two morphological forms. The first one class are the yeast. Now, the yeast class are single cells. When you say single cells, again, they are known as unicellular. They are usually spherical to ellipsoidal in shape. Pag tiningnan mo siya under the microscope, if you look at this under the microscope, sometimes you could mistake this for a WBC or an RBC. And one way, class, for you to differentiate them is that you would be able to identify them based on their budding characteristic. If tiningnan mo tong cell na to, this is an example of your budding yeast cell. As I mentioned kanina, this would be your mother cell and this would be the daughter cell. So if you notice, that's how they reproduce asexually on their own. They would produce a daughter cell, and between this line, there would be a so-called polar budding scar. So, ayan siya. They are very small class with a diameter of 3 to 15 micrometer. Unicellular, and when you grow them on a media, if you want to see their colony, yeast colonies are typically creamy, resembling bacterial colony. Now, the second morphological form are the molds class. Now, molds would grow as filamentous. When you say filamentous, if you try to put this in another synonym, they are branching. So if you take a look at the picture on the lower right, yan, these are examples of your molds. They tend to produce this filamentous or branching tube-like structures that are known as your hyphae. So itong nakikita nyo, this root-like, this branching structures, tube-like structures, these are known as your hyphae. That can vary in a diameter of 2 to 10 micrometer. They are multicellular with cottony mycelial mass. Now, a hyphae, as mentioned, is the root-like, rod-like structures that are unique to mold. So, meaning class, they can only be found. Hyphae can only be found in molds. There is no hyphae for yeast. Now, take note that a group or a mass of hyphae would form a so-called mycelia. So, all of this, itong group of hyphae na to, this is now known as your mycelium or mycelia. Take note of that. Now, how would you differentiate yeast versus molds? Now, there are three ways to differentiate them. You could differentiate them based on the structure, based on the appearance, based on the method of reproduction. Take note, class, that yeast are unicellular when it comes to their structures, and they have buds growing on them. So they are able to perform budding. While molds naman, class, are multicellular with tubular filamentous hyphae that would appear as branches. So that's one way to differentiate them. Yeast, unicellular with buds growing on them. Molds, multicellular, tubular, filamentous hyphae. Appearance of yeast class are round or oval shape, while molds would appear thread-like. Method of reproduction, take note, is that yeast are able to reproduce via budding and in some cases able to perform binary fission. But this is a very rare type of fungi class. More on budding ang yeast nyo. While your molds can either produce sexually or asexually. So do take note of that. Now let's talk about the different types of your hyphae. Now, the first type of your hyphae is your vegetative hyphae. When you say vegetative hyphae, they penetrate the media and absorb food. Now, for you to understand, take a look at this picture on the lower center. Imagine this would be your media. Let's say you use blood agar plate. Upon inoculation, you let the fungi grow. It would now produce hyphae that would penetrate from the bottom. So pupunta siya dun sa ilalim, class. It would go down or it would penetrate the lower part of the media, resembling roots. So the thing you need to remember about vegetative hyphae is that they are like hyphal roots. Mga roots sila, class, and they would absorb food. While your aerial hyphae naman, so on the top of the media, this one. This is the reproductive hyphae or the aerial hyphae. This is directed above the surface of the media. The third one, the reproductive hyphae naman, this would carry the reproductive spores. Now, if we want to compare this to a plant or to a tree, para siyang puno. So, kunwari ito yung puno, sorry, ang pangit ng drawing. So, ito yung tree nyo. This would be the vegetative hyphae, yung roots. They would penetrate the media and absorb the food. While on the center, this would be the trunk. The trunk class would also be known as your is your aerial hyphae. They are directed above the surface of the media. And the last one, the last type of hyphae is the reproductive hyphae. This is like the branches class that would contain the spores. Itong spores na to, if we try to compare this to plants, they are like pollen. So once these spores are inhaled, once these spores are transferred to another media, they could reproduce. So that's the thing you need to remember about the spores. They when they are inhaled or when they are scattered, they are able to reproduce depending on the location on where they went to. So I hope that was clear. Let's continue. Let's talk about the different structural appearance of your hyphae class. So, okay, this one. There are four types of hyphae or four structural appearance of hyphae. The first one is your septate. Now, when you say septate, they are with cross walls or septa. So, imagine this is your hyphae. If you see cross walls or basically parang layers class, they are known as septated. But if they are without cross walls, as in smooth, clear siya, clear yung hyphae, it would be considered a septate. While the third one, the third type, is hyaline. When you say hyaline class, these are transparent hyphae. There is no color. No color siya class. And if you try to stain them, it can absorb the stain. And the last one is your dematiaceous hyphae, which is a pigmented hypha. So let me repeat: if it's septated or septate hyphae, this has cross walls. If it's a septate without cross walls, if it's hyaline, it would be transparent, no color, and if you stain it, it can absorb the stain. And if dematiaceous pigmented hyphae, it will be color. So another recall class: when many hyphae are joined together, they form the so-called mycelium. So before we continue, let's test if you understand the structural appearance of hyphae. Let's go back to one picture. Okay, here, this one. Balik tayo dito, class. Okay, can anyone tell me, or can anyone tell or describe the hyphal structure? This one, ito na lang. Itong sanga na to, class. Itong hyphae na to. Can anyone describe me the hyphae of this mold? Okay, so the answer for this class, this is an example of a transparent, or sorry, not transparent. This is an example of a hyaline septated. It is septated. Why septated class? Tingnan nyo to. There is a cross wall. So may mga cross wall siya. This is a hyaline septated hyphae. So I hope that was clear. Now let's talk about the parts of your mycelium. So as mentioned, when your hyphae are joined together or are grouped together, they form a mycelium class. Now your mycelium has two parts, the thallus and the aerial mycelium. The thallus class again would refer to the vegetative part. It absorbs water and nutrients. So parang ito yung roots. It just has a different term. Thallus ang tawag if it's a mycelium. And then we have the aerial mycelium, reproductive part containing the fruiting bodies that produce the reproductive structures known as your conidia or spores. So do take note of that. Kasi yung fruiting body is the one that is able to reproduce, or the reproductive structures are known as the conidia or the spores. Now let's talk about another type of fungi, the one that is most deadly, class. Ito yung most deadly to humans. The variable fungi. When you say variable, they can change. Fungi that could change. Now, when you say change, these are fungi that can transition between a yeast and a mold. So, kaya niyang magbago. The fungi could change from a yeast to a mold, or from a mold to a yeast. Now, the variation of shape is directly related to pathogenesis, since different forms may be better suited for different microenvironments. Now, for you to understand this statement, your variable fungi, or your another term for variable fungi, pakinote ito, kindly write this down, is that they are also known as your dimorphic fungi. Now, your variable fungi or your dimorphic fungi, as mentioned, can change between yeast and mold. Now, remember, our body, class, has a body temp at the most of 37 degrees centigrade. Now, when fungi, class, wish to invade our body, they would usually be in the yeast form. Now, you may be wondering, sir, why is it in the yeast form, class? Your yeast, class, would survive at an optimum, kindly write this down, at an optimum temperature of 37 degrees centigrade. While your molds, class, has an optimum temperature of 20 to 25 degrees centigrade. And this 20 to 25 degrees centigrade, in different books, so about 20 to 30, but let's use the 20 to 25. For 20 to 25 degrees centigrade, this is also known as your room temp. So ang yeast nyo, class, they have an optimum temp of 37 degrees. So if the yeast wants to serve, or if the fungi wants to survive in our body, they need to be in a yeast form. Kaya most of the time, if you have a type of fungal infection, what you would isolate in the body, kindly write this down. What you can isolate in the body, class, would be the yeast form of the fungi. Rarely kayo makaka-encounter ng molds inside the body because they cannot survive. They can only survive at 20 to 25 degrees or 20 to 30 degrees, or basically the room temp. That's why, as mentioned in the statement, the variation is dependent also, class, on their environment. So do take note of that. So these are the different variable fungal forms. So if it's a monomorphic fungi, it only has one form, has only one growth phase. It can either grow as a yeast or a mold. If it's dimorphic fungi, at room temp, class, take note of this, ito yung ina-explain ko. At room temp, they would grow as molds. But in a body temp, especially in tissues, they would grow as yeast. And we also have a diphasic fungi. This is the one that's capable of changing instantly, class. Pabago-bago siya. Kaya niyang magbago kagad depending on the temp. So it could be a mold at room temp, then magiging yeast siya in 37 degrees centigrade. So bigyan ko kayo ng example dito sa laboratory. Let's say you tried to isolate, or you isolated fungi from your CSF. Now you put them on two plates. One plate would go to the incubator, 37 degrees, di ba, kapag incubator. One plate would go to a room cabinet. So room temp siya. Upon noticing, both would have positive growth. Both would have positive growth. From there, you could already identify, class, that the fungi you isolated is dimorphic. Now, how would you differentiate diphasic? Kapag naman diphasic, let's say, you took, kinuha mo to, you took the one that was stored at the incubator at 37 degrees centigrade. Then you decided to store it in a, or wait, ibahin ko, class para mas ma-clear. You took you took a isolate or a colony from the 37 degrees. So from the yeast colony. And then inoculate mo to. You decided to inoculate this to another media. This time, you decided to put it on a room temp. If there is a positive growth on the room temp, this is now considered a diphasic fungi. Kasi the colonies you took from the 37 degrees or yung yeast na kinuha nyo, the yeast you took from the 37 degrees was able to grow at the 25 degrees centigrade, becoming mold. So I hope that explanation was clear. To simplify, class, kapag dimorphic, they can both grow at room temp and at 37 degrees. Pag diphasic, they can grow in one temperature. Then they could also, if you try placing them in a different environment, they can also survive. So I hope that was clear. Now let's talk about the methods of reproduction for your fungi. Now, fungi may reproduce either through sexual or asexual reproduction. Now, let's talk about the two types, sexual and asexual. Kapag sexual, class, this would require two haploids. Pero pag asexual, sarili lang niya, self lang. So let's talk about the first one, the anamorphs. The anamorphs, class, from the word A, para matandaan nyo so that you could remember, anamorphs is the asexual form. A is to A. Yan yung way tandaan nyo siya. Like anamorph, asexual. And its reproductive elements are known as conidia. So yung they reproduce via this conidia, class. They would usually involve mitotic division of the haploid nucleus and is associated with production via budding. So yung single haploid nucleus, class, magproproduce siya on its own through the so-called budding, which are producing spore-like conidia or by the separation of certain hyphal elements. Then the second one is your teleomorph. Now, teleomorph, class, is the sexual form, and its reproductive elements are known as your spores, such as your ascospores, zygospores, and basidiospores. So let me repeat. If it's anamorph, it's the asexual form. Its reproductive elements are termed conidia. They divide by mitotic division, wherein the haploid nucleus would divide on its own via budding, producing spore-like conidia or separation of hyphal elements. If it's teleomorph, it's the sexual form, and reproductive elements are called spores, known as your ascospores, zygospores, and basidiospores. It would involve the haploid nuclei of donor and recipient cells. So, in sexual form, as I mentioned, it would require two cells, a sperm and an egg in mammals, in this case, two haploid nuclei, and they would fuse to form a diploid, which then divides by classic meiosis. So let's talk about the different spores or the different conidia involved in asexual reproduction or the anamorph. The first one class is the conidia. These are spores produced singly or multiply in long chains or clusters by specialized vegetative hyphae known as conidiospores. And this conidia class comes into two types. They can either be a macroconidia or a microconidia. From the prefix macro, the prefix micro, like sinabi mong macro, these are large and they tend to be multicellular. If it's microconidia, they are small and they are unicellular. So this is an illustration class of what a conidia would appear. So sabi dito, they can produce singly or multiply in long chains of vegetative hyphae known as conidiophore. So this one, this hyphae class is known as your conidiospores. And they can produce either a microconidium or a macroconidium, either singly or multiple. So yan siya. So macroconidia would be a large conidia, multicellular. Kaya siya multicellular class, they tend to have groups of cells together. Yan siya. Yan. But it's microconidia, small, and unicellular. Let's go to another spore involved in anamorphs, the blastoconidia, also known as the blastospores. They develop as daughter cell buds off the mother cell and is pinch up. So ito yung, to simplify this is an example of a yeast cell that undergoes budding. So ayan siya. So this is the blastoconidia wherein a daughter cell, ito yung daughter cell niya class, would bud off from the mother cell. The third one is your chlamydoconidia, also known as your chlamydospores. They are thick-walled, resistant resting spores produced by rounding up and the enlargement of a hyphal part. Now, for your chlamydoconidia or chlamydospores, there are three locations. It is known as a terminal chlamydoconidia if it is located at the end of the hyphae. It is known as an intercalary chlamydoconidia if it is found within the hypha, sa loob ng hyphae class. It is a sessile chlamydoconidia if it's found at the side of a hypha. So, for you to understand, ganyan siya. Take a look this class that these are thick-walled. So, nanonotice nyo that the cell wall is thick, makapal. And they tend to produce by rounding up and enlarging the hyphal part. So, yung hyphal part would enlarge class. So, and there are three locations. If it's found at the end of a hypha, it is known as a terminal chlamydoconidia. If it is found inside the hypha, it is known as intercalary. And if it is found class at the side of the hypha, it is known as a sessile chlamydoconidia. So take note of the three locations or the three types of chlamydoconidia based on the location. It's a terminal, intercalary, or sessile. They are thick-walled, resistant resting spores, and they would typically enlarge the hyphal part. The fourth type of anapomorph or asexual conidia is your arthroconidia, or also known as your arthrospores. The thing you need to remember about arthroconidia class is that they involve the fragmentation of the mycelium. Pag sinabi mong fragmentation, they tend to disjoint. Mag-detach sila class. They would tend to detach That's why when they appear class, they can appear as jointed. Parang may mga harang or there would be walls. They are rectangular or barrel shaped spores. So ito yung tsura. So take a look at the appearance class. There is a fragmentation. If you take a look at the hypha, they are fragmented or wasak-wasak sila or kalat-kalat sila. They are separated from one another. Then they would appear as jointed. So there would be joints together, may mga parang spacing in between. So that's the correct term. There would be spacing class in between the spores. And they tend to have a rectangular or barrel shape appearance. So that's the arthrochonidia. The fifth one is your sporangiospore. So the fifth type of anamorph is the sporangiospores or the sporangioconidia. These are spores contained in a sporangia. So meron siyang parang sac, SAC class. When you say sac, parang container that are produced terminally on a aseptate hyphae known as the sporangiospore. So this is the appearance. So this is your aseptate hyphae. If you take a look at the hyphae class, there is no cross walls. And then it is produced terminally. So at the end. Di ba pag sinabing terminally, located at the end. And there would be a sac-like sporangia. So itong nakikita nyo, this one class, this balloon-like structure that covers the this is known as your sporangia. And inside that sporangia, you can find the sporangiospores. So yung nakikita nyo, the one you find, the small circular structures inside, that's the sporangiospores. Now let's go to the teleomorph or the sexual reproductions. We have the ascospores. So do not mix together the arthrochonidia class. Iba yung arthrochonidia, iba yung ascospores. So if it's ascospores, they are the one involved in sexual reproduction. They are spores class contained in a sac-like structure known as your ascus. So kung sa sporangiospores, meron siyang sporangia. Dito sa ascospores, there is the so-called ascus. So, ayan siya. So this is your spores class. Yeah. They look para siyang no? This looks like a hookworm. Kala mo parasite. Anyways, so inside this ascus class, ito yung covering or yung sac-like structure. This is the ascus. And inside the ascus, you can find the ascospores. So that's your ascospores. The second one would be your zygospores. Involves the fusion of two identical cells arising from the same hyphae. So fusion of two identical cells arising from the same hyphae. So ito siya. For you to understand, this is the hyphae class. What happens is that one cell would grow here at this side and another fungal cell would grow here at the side. They would bind together or they would combine, forming a zygospore. Same hyphal structure sila. Sige, tingnan nyo. This is the hyphae. Single hyphae lang to class. Around the same time, both of them, or within the hyphae, two cells fuse together, forming the zygospores. So zygospores are two identical cells arising from the same hyphae. The third one is the oospores. Now, the oospores naman class is a fusion of cells from two separate non-identical hyphae. So, magkaiba naman ngayon if it's oospores, different or non-identical hyphae. So, ito siya. So, this is one hyphae class. This is one hyphae, they fuse together, forming the so-called oospores. So, para matandaan nyo lang siya, yung isa non-identical hyphae, yung isa identical hyphae. The fourth one would be Basidiospores class. So this is a spore in a club-like or in a club-shaped basidium. So this time naman, yung sac niya or yung container niya is club-shaped. So para ma-appreciate niyo yan. Ayan siya. So inside this basidium, ito yung basidium nyo. If you take a look, club-shaped siya, di ba? Inside the basidium, you would find the basidiospore. So, ayan si basidiospore nyo. They are found inside the basidium or the club-shaped basidium. Let's talk about the mode of transmission for your fungi class. Now, they are usually acquired from the external environment via the following. The first and the most common way you can acquire a fungal infection class is via inhalation of the reproducing body known as your codidia, usually from environmental modes. Some are ubiquitous. When you say ubiquitous, they are found everywhere. But there are some fungi class that are restricted by climate that favors their growth. So some fungi cannot grow in a cold temp in less than 10 degrees centigrade. Most fungi cannot grow. Yung mga molds nyo, they cannot grow at 37 degrees. The same for yeast. Yeast cannot grow in body temp. Body temp. Uh, Room temp. They cannot grow in room temp. Molds cannot grow in 37 degrees centigrade. So there is a restriction. That's why if you're familiar with the movie Last of Us class, 'di ba yung fungi doon, the fungi was able to adapt from surviving in forests. So pag nasa forest yan, most likely humid yan. It would be around 20 to 30 degrees centigrade. So from there, it was able to adapt to the human's body temp, yung cordyceps, the cordyceps fungi. So hindi natin siya i-discuss class kasi hindi naman siya relatively important to humans. But if ever there's a possibility in the future, hopefully wag naman, and it would affect humans, the same pathogenicity or the same mode of infection niya wherein it would control the brain of its target, that would be a big problem for humans. Kasi fungi class, imagine inhalation. So simply by inhaling, you could get infected. Now, another common problem with fungal infections among bacteria, viruses, and fungi. Fungal infections are commonly found in immunocompromised patients. Now, if you're familiar when we talk about immunocompromised patients, these are HIV positive patients class. Those have developed AIDS and they have a very weak to none immune system. That's why most of the time ang first disease ng mga may HIV class are usually fungal diseases. Manonotice nyo na lang yung balat ng patient, may ganito na, yung tissues niya, may ganito na. They're very common among immunocompromised patients. So, yan. And they can also be acquired via tissue penetration and invasion. So, when we talk about tissue penetration and invasion, we're talking about how they could get into our body via abrasions, Yung mga nakat ka, nasugat ka by cuts or penetration basically. So that's how fungal would get into us. Inhalation from immunocompromised patients, they can survive in immunocompromised patients, and there would be tissue penetration and invasions via abrasions and cuts. Now let's talk about specimen collection class. So for specimen collection, if you're going to collect skin specimens, so you're going to inoculate or you're going to acquire skin specimens, it should be cleaned first with 70% alcohol. The reason for cleaning it with 70% alcohol is to remove dirt, oil, and surface saprophytes. When you say saprophytes class, these are fungal contaminants. Now, same procedure must be done, class, if the specimen is a nail. So, if it's a nail, you need to clean it first with 70% alcohol. And after that, it should be clipped and excuse me, it should be finely minced before inoculating to media. Now, if you want to collect hair, it can be done by plucking, brushing, or with a sticky tape. And for a normal sterile procedure must be done if the specimen is a body fluid. So if it's a sputum, etc. So when you say normal sterile procedure class, make sure that you gargle first. If you want to collect sputum, gargle ka muna, you gargle first, then you collect. If it's a body fluid, make sure you clean it first with alcohol, the site of puncture, alcohol, iodine, and then alcohol. It's the same procedure as if you're trying to collect uh, blood samples. Diba, if may request ng blood CS, you need to clean it like that. Alcohol, betadine or iodine, then alcohol. So, same with all collection of body fluids, you need to clean it like that. Now, how would you transport and handle them? So, for hair and nails, they need to be sent in a dry envelope inside a proper container. Other specimens are usually sent frozen or on dry ice. Specimen must be inside a packaging with a biohazard regulation. Any growing cultures must be on tube media, not in plates class. So dapat nasa tube media siya. The reason why it's in a tube media is because tube medias class have a cap. And the cap class, yung takip, it would seal the media, preventing inhalation of spores. Imagine mo kung nasa plate yan. You just lift the plate, onting galaw sa plate class, you could easily get the fungi. So by sealing it in a tube media, you prevent the inhalation of the spores. It needs to be you need to use an aluminum screw cap with outer cardboard mailing tube is usually the container of fungal specimen. Inside labeling information must contain the patient ID, the specimen source, and the suspected organism. So you need to include the suspected organism class for so that the medtech could properly prepare which biosafety procedure is to be followed. So, susundin niya ba yung biosafety level 3, biosafety level 4, 1 or 2, something like that. So, this is the specimen processing. Kindly take note of them. For the skin, the nail, and the hair, they are directly examined following KOH preparation. KOH class, this is your potassium hydroxide. The reason, class, take note of this, the reason why KOH is used for skin, nail, and hair. So before I answer pala, I have a question. Anyone could answer. Skin, nail, and hair, the three of them, itong tatlong to, they contain What? Anong meron sa kanila? What do they contain that are similar among the three? Ulitin ko. Among skin, nail, and hair, what what do they contain that is similar among the three? So itong tatlo na to class, they contain keratin. And the purpose of your potassium hydroxide is that they can dissolve. It can dissolve keratin. So by dissolving keratin, you can easily find or you can easily look for the fungi. Then for body fluids such as your pleural fluids, sputum, and bronchial aspiration, so these are respiratory samples. For respiratory samples, specimens must be collected in fresh due to possible growth of saprophytes or yung mga contaminants niya over pathogens such as H. capsulatum. H. capsulatum class H would stand for histoplasma. Then for gastric washing, same procedure lang din class. So it needs to be collected fresh. For genito-urinary specimen, for the urine, first morning specimen is preferred. For blood and bone marrow, directly inoculated in your BHI broth or BHI slant. BHI, alam ko familiar kayo dito. This is known as your brain heart infusion. So they come in a broth form and a slant form. Then for wound abscess or drainage, they need to be cultured anaerobically, especially if the disease suspected is actinomycosis. 'Di ba ang mga actinomycetes nyo class are anaerobic. So they should not be exposed to oxygen. For CSF, you need to centrifuge and then examine the sediment microscopically, then inoculate on media. For tissue specimens, examine for pus, gaseous material, or granules. Mince aseptically and can use small sterile saline supernatant once inoculated. Please copy. All right, let's go to the laboratory diagnosis of fear fungi. So fungi can be identified by directly observing their distinctive morphological features on direct microscopic exam of specimen. So medtechs would usually look for spores, hyphae, mycelial elements, budding yeast, and mycotic granules. If you still remember class in your AUBF, ito may question ako. In your AUBF, what fungal element can usually be seen in urine? Yung pinaka common fungal element that could be seen in the urine. So the answer to this, if tama sagot nyo, would be the yeast cells. So most of the time, if a patient has urinary tract infection class and sobrang severe na niya, or even in old or geriatric, yung mga geriatric patients, there's a high possibility for you to find yeast cells. So as medtechs, we would look for these spores, this hyphae, yeast cells, etc. So for diagnosis, we are going to prepare a wet mount preparation. So when we talk about wet mount, we use NSS. We put the NSS in a slide, then the sample, then we add the cover slip. Tapos we view it under the microscope class. So this is good for yeast examination. the loss of fragile structure is minimized. Then we also use 10% KOH mount, good for skin scrapings, hair, nails, sputum, vaginal specimens. KOH would clear the specimen's tissue cells, mucus without destroying the cell wall so that fungal elements could be seen. Then fungal stains could be used class for direct examination. They enhance the visualization of fungal structures. And then the gold standard. So take note of this. Same lang din sa bacteriology nyo. Di ba sa bacteriology, the gold standard for bacterial identification is culture. So for fungi, it's fungal culture. It's the gold standard. Culture is more sensitive than direct examination, and a portion of the specimen used in microscopy should be cultured. So after performing microscopy class, whether may nakita ka, whether you were able to find a fungal element or not, you should still culture it. Culture class must be held for 21 days at room temp. Yeast would grow better at 37 degrees and molds at 30 degrees centigrade. So decision nyo class if you want to put it on a incubator or in a room temp cabinet. So if either grows, kung may nag-grow yan sa incubator nyo, most likely it's a yeast. If it grows at room temp, most likely it's a mold. And among the two, the least pathogenic would of course be molds. The reason, kasi nga 'di ba ang molds nyo, they cannot survive in our body temperature. But if it grows as a yeast in an incubator, kabahan ka, you need to wear the proper personal protective equipment. So mag N95 mask ka, try to implement a biosafety level 3 for fungal culture. As much as possible, you work inside a biosafety cabinet. And you need to hold it for up to 21 days. Diba sa bacterial culture nyo, it should usually last for 7 to 14 days. But for fungal, as long as 3 weeks class. So these are the different fungal stains used. The most common one is your lactophenol cotton blue, or the LPCB. This is usually used as a quick evaluation of fungal structures. It stains the chitin in the cell walls of fungi. Then we have your PAS, or your periodic acid shift stain. Stains the polysaccharide in the cell walls of fungi. Fungi would stain purplish red with a blue nuclei. So sa LPCB class, ang color ng fungi niyo would be blue. But for PAS, ang ini-stain niya is polysaccharide sa cell wall. So they would appear purplish red and would have a blue nuclei. Another stain, yung Gomori methenamine silver stain, or the GMS. Outlines would appear in black due to silver precipitating on the fungal cell wall. Internal structures are deep rose to black, and background is light green. So ang appearance class ng fungi nyo, the cell wall would appear black, the structures would appear deep rose to black. Background is light green. Another stain, the Gridley stain. Hyphae and yeast class would stain dark blue or rose. Tissues would stain deep blue, and the background would appear yellow. Then we have your Mayer musicarmine stain. This would stain the capsule of your Cryptococcus neoformans deep rose in color. Then we have your fluorescent antibody stain. This is a simple, sensitive, and specific, applicable for many different fungi. So sa fluorescent antibody stain class, depending on the type of stain used, most of the time, the fungi would mag-fluoresce siya, yung kumikinang-kinang, mag-fluoresce siya in the color green. Pakitake note of that. They would fluoresce in a green color. Then we have your pap stain, your Papa Nicolau stain, good for initial differentiation of dimorphic fungi. Works well on sputum smears. So yung pap stain nyo, you will learn about this in your histopath. There are a lot of uh, pap stain has a lot of stains used or chemicals used. Then we have your gram stain. So when you perform gram stain, yung crystal violet nyo, yung iodine, yung safranin, most fungi class are usually gram positive. So kapag gram positive siya, they would appear violet or purple. Then gem sustain used on blood and bone marrow specimen. So if you think or if you have a have a initial suspect that the patient has extensive fungal blood infection, you can use gem sustain. So gawa kayo ng blood smear class. Gawa kayo ng blood smear. Then you use gem sustain. But again, very rare mangyari yan. In my practice, I have never encountered a yeast surviving in blood. Then we have India ink. So pakitandaan tong India ink na to class. It demonstrates also the capsule of Cryptococcus neoformans in CSF specimen. So pakitandaan siya. Please copy. Now let's go to the fungal culture media. So the first fungal culture media is the birdseed agar. So this is the isolation and preliminary identification of Cryptococcus neoformans. Sa birdseed agar, the colonies would appear as black colonies. Then we have your cornmeal agar, usually used on culturing chlamydospore bearing Candida albicans. So this is used for Candida albicans class for the chlamydospores that are produced by the Candida. Then there's also cornmeal agar, but this time there's an additional nutrient, 1% dextrose. This is used to improve the pigment production to differentiate Trichophyton rubrum from Trichophyton mentagrophytes. If you try to grow them in cornmeal agar with 1% dextrose, Trichophyton rubrum would produce the red pigment, while mentagrophytes does not produce red pigment. Then we have your Sapex agar used to isolate Aspergillus species. Itong Aspergillus species nyo class, this is a very common contaminant. Most of the time, you can find this in your bathroom walls sa mga tiles ng banyo nyo. They have different appearances or different species. Merong Aspergillus niger. So kapag nakakita kayo, if you find black molds in your bathroom CR, most likely Aspergillus niger yan. So that's one ano. So you can use Sapex agar to isolate Aspergillus species. Madaming species ang Aspergillus class. It's a very common contaminant, a very common saprophyte. Then we have your DTM or the dermatophyte test medium, usually used to isolate dermatophytes. So in the next lesson, you will learn about dermatophytes class. So used for isolating dermatophytes from cutaneous specimens. Then we have your mycocell or mycobiotic agar. So mycocell or mycobiotic agar class, they are selective. So pag selective class, they would usually grow a specific or an isolated organism. So they are used to isolate pathogenic fungi, dermatophytes, and systemic fungi. They contain different antibiotics class, such as cyclohexamide. See, cyclohexamide, they would suppress the growth of saprophytic fungi. So, sinosuppress niya yung mga contaminant fungi. And for chloramphenicol naman for bacterial contaminants. So if asked in your quiz what suppresses saprophytic fungi, yung antifungal is cyclohexamide. Pag bacterial contaminants naman, chloramphenicol. That's for mycocell or mycobiotic agar. And the most common, so ito class, this is known also as, kindly write this down. This is the routine media. Kung meron tayong nutrient agar or nutrient broth sa bacteriology, in your mycology or in your fungi, we have the so-called saborodextrose agar or the SDA. They contain glucose, modified peptone set at a pH of 7, supports growth of fungi, and restricts the growth of bacteria. And these are the different texture and color of colonies class. So most of this, if you take a look, this is how you would appreciate the different parts of the hyphae, from the vegetative, the reproductive, and the aerial. So imagine if it's a hairy colony, most likely class molds to. These are the mycelium. Now you would have the thallus and the vegetative, and if you take a look at in a hyphal appearance, makikita nyo nandito sa taas yung mga reproductive spores niya or reproductive conidia niya. Andyan din yung vegetative hyphae niya below the media. So in determining the color of colonies, you need to look for two colors class. The color of the margin. Pag sinabi mong margin, you're talking about the outer appearance. So yung outer layer niya. Then the color of the center. Then observe for grooves or cuts. So itong nasa labas, outer layer niya or outer circular appearance niya, and also for the center. Look for grooves or cuts. Yan. Ito, para mas maintindihan niya. Ito, margin color, then center color. Then look for groups or cuts. Now let's discuss miscellaneous fungal tests. We have the so-called germ tube test. Now the germ tube test class is a very important initial test in the identification of yeast isolates. So sa germ tube test class, gagamit kayo ng serum. Then you inoculate your fungi, especially yeast. Then after that, i-incubate nyo siya. If you find budding, mga nagbabudding na yeast cells, positive siya for Candida albicans. So ulitin ko ha, serum, you inoculate it with fungi, especially yeast, incubate it at 37 degrees centigrade or sa incubator. Then if you find budding yeast cells, it's positive for Candida albicans. Siya yung confirmatory test class. It's the confirmatory test for Candida. Then we have your L-DOPA ferric citrate test used in the rapid identification of Cryptococcus neoformans. So si Cryptococcus neoformans is capable of using phenol oxidase. Positive color is black. So yan siya. This is the appearance of your yeast cells, Cryptococcus neoformans, if positive for L-DOPA, color black. Then we have urease test. A positive result is alkalinization of media indicated by a pink color. So ang positive result ng urease test nyo is pink color. The reason is because the media turned alkaline. Positive for Cryptococcus. It can also be used to differentiate T. mentagrophytes, Trichophyton mentagrophytes from Trichophyton rubrum. So si Trichophyton mentagrophyte is urease positive. Si Trichophyton rubrum is negative. Then you have your hair baiting test. Differentiates pentagrophytes from rubrum. Sorry, typo. So si pentagrophytes positive, si rubrum negative. There would be V-shaped penetration of the hair shaft. So ayan siya. When you do the hair biting test, if it's positive for mentagrophytes, the stain would penetrate the hair shaft. Because inside the hair shaft class is your fungi. So yung fungi na mentagrophytes would be stained. While if it's negative, there would be no penetration. So that's hair biting test. Other important stains and mounts. So this is differentiated into two types class. Let's talk about the first one, yung temporary mount. Pag sinabi mong temporary mount, when we talk about temporary mount, these are substances or fluids added to the slide. So mga substances lang to class, katulad ng NSS. So these are mountants. So, the first one is 10 to 20% KOH. Dissolves tissue material, so fungal elements may be more visible. Dissolves keratin. This is a rapid method of detecting fungal elements. Gentle heat class increases the rate of clearing. So, ang ginagawa nila in some laboratories, after adding the KOH, they would pass it in a flame. Dadaan-daanan niya sa flame class so that mas mabilis yung pag-clear or pag-dissolve ng keratin. Then we also have your lactophenol cotton blue, LPCB, preserves and stains fungi. Excellent mounting media for fungi. In the future, if you're going to work in a hospital class, the most common stain and mountant that you would encounter for fungi is these two, KOH and LPCB. Then another stain is your India ink, also known as your nigrosin, for Cryptococcus neoformans. Then we also have your calcofluor white stain. Si calcofluor white stain yung class, this is fluorescent, a fluorescent dye or a fluorescent stain. Usually used in FM microscopes or fluorescence microscope. The elements would typically appear bluish white against a dark background. Earlier I mentioned, di ba, in some stain or fluorescent stain, they would appear green. But for calcofluor white stain class, the fungal element would fluoresce bluish white in a bluish white background, in a bluish white color or in a bluish white fluoresce against a dark background. Then we also have another type of mount, the permanent mounts. So the first one would be gram stain. So particularly useful for the detection of yeast. Most yeast would stain partially or completely gram positive. And they are usually differentiated from bacteria due to their large size. So, di ba ang bacteria nyo, your bacteria can only be best viewed in oil immersion objective. But for yeast, molds, HPO, pwede na class. So they tend to be larger in size. Then we also have your GEMSA or right stain. So this is used in cases of histoplasma. Demonstrates yeast cells within macrophage. If you remember, di ba, your macrophage class is capable of phagocytosis. So what it would do is that it would engulf histoplasma. And using GEMSA or right stain, the macrophage or the monocyte, you could view the histoplasma. Then periodic acid shift stain, fungal hyphae would be PAS positive. Then gomorium methanamine stain has a high contrast with minimal background staining. Hematoxylin, hematoxylin, and eosin, also known as the HNE class. This is very important for hyphae class because it determines if the fungi is hyaline or colorless, or dematiaceous or pigmented. Then we have Mayer's Musicarmine, mucoid capsule of Cryptococcus, Fontana Mason to identify dematiaceous or pigmented fungi, acid fast or quinions to differentiated Nocardia from Actinomyces. Si Nocardia nyo class is slightly acid fast. And the last stain is your acridine orange, useful in detecting Tinea versicolor. Would have a green fluorescence of the fungal elements. Please copy the last one class, yung acridine orange. I forgot to include that in the handouts. Okay, so that basically ends class the first lesson for mycology, the introduction to mycology, and these are the different references used in creating this PowerPoint. Thank you.